Welcome Genetic Teacher Channel. Today, we are talking about loading dye through agarose gel electrophoresis, which can be done by mixing bromophenol blue dye with DNA samples to be loaded into a gel after the tank has been filled with TB buffer. So, loading dye contains a dense substance, such as glycerol, to allow DNA samples to fall into samples wells as a tracking dye, which migrate in the gel and allow monitoring how far the electrophoresis processed. These process will be done by adding loading dye to all DNA samples onto the agarose gel in approximate 100 voltage for 30 minutes to monitor migration of DNA samples, as mentioned inside slide. This slide showed the typical agarose gel with time. As we showed, the DNA fragments are not visible on agarose gel electrophoresis without loading dye. In abbreviate, agarose gel separates DNA fragments according to size, and the electrical current carries negative charged DNA through gel to positive electrode, hence, small fragments move faster than large fragments. Thanks for your attention. Please like and share and subscribe our videos and channel.